Streptococci. These are the group of organisms that are mostly involved in the pyogenic infections. So similar to that of the staphylococci, these organisms are also gram-positive in nature. And the difference between the staphylococci and the streptococci is that staphylococci usually causes localized infections, whereas the streptococci has the ability to cause systemic infection because it has a nature of spreading into the body with the help of different enzymes that it produces. So coming to the introduction part of this streptococci, these organisms are gram-positive, that is they stain as violet in gram staining, and they are cocci in shape, arranged in chains or pairs. Usually they form linear chains in contrast to the staphylococci, which are in bunch form. So these organisms divide and in one plane and form chains of organism, and hence they are called as streptococci. And most of these organisms are the one that are the normal flora of the human body. That is, they are normal inhabitants that are present on the human body. But given an opportunity, these organisms has the ability to cause the infection, and hence they are called as opportunistic pathogens. And sometimes some species of these organisms are able to cause lethal pyogenic infections, and most of the organism which causes these infections belongs to Streptococcus pyogenes. Hence they are called as pyogenic infections. Pyogenic infections are nothing but they are the one that usually forms pus in the wounds or the burns, and can cause a sequel of infections such as rheumatic fever and the glomerulonephritis, that damage the kidney cells. And these organisms spread from one part of the body because they have the ability to produce certain enzymes called as streptokinase that breaks the fibrin clots and helps the organism to spread the infections. And they were first isolated in were said to be related to the non-suppurative lesion organisms. That is, they were not supposed to cause pyogenic infection, but later on when these organisms were isolated from the acute abscesses, they were found to be involved in the suppurative infections. And apart from the suppurative infection, they also have the ability to carry out a sequel infection. That is, after the acute infection from the streptococci, a sequel to this infection is followed, which is via the acute rheumatic fever or the glomerulonephritis. These organisms were first isolated in the patients of who were suffering from erysipelas, that is a skin rash, by Bill Roth, and he called them as streptos. That is, they are called as twisted or coiled organisms. Next, these organisms were also isolated from the acute abscesses by Oxton, who also isolated the organism staphylococci, and he distinguished this from the staphylococci because staphylococci are mostly involved in the localized infection, whereas this has the ability to spread to different parts of the body. And Rosenbach was the one who isolated the organisms from the human suppurative lesions, and he named them as Streptococcus pyogenes because these organisms were pyogenic in nature. Now coming to the classification of these organisms, there are many species related to these organisms that can cause infection not only in humans but also in the animals and other organisms. So these organisms, Streptococci, are mostly divided into two major types, that is aerobic and the facultative anaerobes, and the other one is the obligate anaerobes. The group of the organism which are Streptococci or obligative anaerobes are the Streptococci, which we'll be dealing later. To this particular topic, we'll be studying about aerobic and facultative anaerobes. As we all know, aerobic organisms are the one that requires oxygen for their growth, and facultative are the one that can survive even in the minute presence of the oxygen, whereas obligate organisms are the one that are strict in their oxygen requirement. That is, they don't require any amount of oxygen to survive. Even a small amount of oxygen can kill this organism. So these Streptococci are the organism that belongs to aerobics and facultative anaerobes. Now the aerobic and the facultative anaerobic streptococci can be again classified based on the hemolytic patterns in the blood agar. So these organisms, because they are pyogenic in nature, they are strict in their nutritional requirement and they grow very well in the blood agar. So based upon the type of pattern of hemolysis they produce on the agar, they can be classified into three major groups. One is called as the alpha hemolysis, beta hemolysis and gamma hemolysis. So respectively the organism can be called as alpha hemolytic organisms, beta hemolytic organisms and the gamma hemolytic organism. And the differentiation or the differences between these hemolytic pattern is that alpha hemolysis usually forms green incomplete lysis of the RBCs, that is they show greenish discoloration in the plate of the media containing blood agar. So most of the streptococci organism which belongs to viridans group are the one that causes the alpha hemolysis. So the alpha hemolytic organism can be further classified based on the physical and the chemical compositions or the biochemical properties. Next, coming to the beta hemolytic organisms, they are the organisms which are pyogenic in nature. So beta hemolysis is the complete lysis of the RBCs that can be seen, and the complete lysis of the organism can be identified as a zone of clearance on the blood agar media. And most of the pyogenic organisms are the one that belongs to the beta hemolytic patterns that they create on the blood agar. The beta-lytic group of organisms causes a complete clearing of the zone. They causes complete lysis of the RBCs. And these beta hemolytic organisms are further classified by Lansfield based on the C carbohydrate that are present on the cell wall of the organisms. And there are different types of streptococci of beta hemolytic, and they are further classified based on the cell wall protein which are called as MTR protein into different types, that is 1 to 8. Coming to the gamma hemolysis, where there is no lysis of the RBCs that can be seen on the media. So these streptococci which causes incomplete lysis or does not cause any lysis are also called as the indifferent streptococci, and they are again divided based on their physical and the chemical properties. Now coming to the properties of alpha hemolytic, beta hemolytic and gamma hemolytic, and this was seen on blood agar containing 5 to 10% of the horse blood agar, and the pattern of the lysis was seen, and the organism were classified respectively based upon the pattern of lysis they created on the RBCs or on the blood agar. So alpha hemolytic organism, the characteristics of alpha hemolytic organisms are they causes green discoloration on the media, and it is called as a partial hemolysis, and the zone of lysis is very small, that is it is about only 1 to 2 mm and with indefinite margin. And most of these organisms that belong to alpha hemolysis are the normal commensals or the normal flora of the human body. Commensals are the one that lives in collaboration with the host, that is the host providing certain components whereas the organism providing in turn giving certain functions to the host. So most of these alpha hemolytic organisms are the normal commensals of the host, and given a chance these organisms will have the ability to cause the infection, and hence they are also called as opportunistic pathogens. Examples of the organisms are pneumococci, especially the streptococcus pneumoniae, or the viridans group, that is streptococcus viridans, which are miscellaneous organisms of the streptococci. Next, coming to the beta hemolytic, here in this form of hemolysis you can see a sharp defined clear zone of hemolysis of about 2 to 4 mm wide, and here organisms completely lyses the RBCs that is present in the media, and hence a clear zone is seen around the colonies. And most of these organisms are pathogenic in nature, and the pathogenic streptococci belong to beta hemolytic group of organisms. Coming to the gamma hemolysis, here you cannot see any change in the medium, and they are referred to as indifferent because they are not able to cause any lysis of the RBCs, and most of these organisms includes the fecal streptococci, also called as the enterococci. So initially enterococcus organism were included under streptococcus, but now they have been classified in a different genus because they have got certain properties which belong to their own but not to the streptococci. So here you can see on the slide how the lysis is usually brought about. In the beta hemolysis you can see a complete zone of clearance, complete lysis of the RBCs which shows clear definite margins around the colonies, and in the alpha they causes greenish discoloration, whereas the gamma is there is no change in the lysis of the RBCs. And further, the beta hemolytic organisms were classified by Lansfield based upon a specific carbohydrate C protein that is present in the cell wall of this beta hemolytic streptococcus. So the nature of the carbohydrate C antigen present on the cell wall helps it to classify further into different groups. So they are called as Lansfield groups, and 20 different Lansfield group of beta hemolytic streptococci are known, and most important one that we'll be studying here is the Streptococcus pyogenes, and these organisms are the one that causes the human infection. So among the different group of beta hemolytic, that is A2, they are nearly T, excluding the I and J. So all these are the one that are involved in the human pathogens, or they are called as a human pathogen, or related to the pathogenesis of the streptococcal infections. And the most important one that belongs to this beta hemolytic group are the Streptococcus pyogenes. And this can be further divided based upon another important component on the cell wall, proteins called as MTR protein. And based on this, Streptococcus pyogenes are divided into 8 types. So first we have the classification based on the hemolytic pattern into alpha, beta and gamma, and beta hemolytic organism further being classified by the Lansfield group into A to T, excluding I and J, and again the Streptococcus pyogenes being classified based upon the cell wall protein, that is the MTR, into 8 different types. So this is how the organisms of there are innumerable number of the genus or the organisms belonging to the beta hemolytic group. So they have been classified based upon different components that are present on the organism. Now we'll be studying in detail about the human pathogenic group of organisms, that is the Streptococcus pyogenes. First, coming to the morphology of the organisms, that is the basic character that we should know about the organism, how it looks under the microscope. So here the organisms are spherical or oval in nature, measuring about 0.5 to 1 mm in diameter, micrometer in diameter, and these organisms are mostly are the one that can change their characteristic depending upon the culture media where they are usually growing. So in the aerobic culture, they show a normal type of growth, whereas in anaerobic culture medium, they are usually smaller in colonies. And depending on the conditions again and the nature of the cultural conditions, the type of change that they are forming also differs. Some Streptococcus pyogenes form short chain colonies, whereas some will form long chain colonies. And here the division of the organism is in single plane, and the daughter cell that is formed remains attached to the parent cell, and hence a long chain of these organisms are formed. And here the longer chain is mostly seen in the liquid medium than in the solid medium. Now, initially it was the length that was related to the virulence of the organism. So the longest one that chains that are formed by this organism belong to the group of Streptococcus salivarius, and depending upon the chain length, they were classified as brevius and longus. And later on it was found that there was no relation between the pathogenesis or the virulence of this organism to the length of the chain, and hence they have been that type of classification that is related to the virulence was discarded. So now they are mostly classified based on the hemolytic pattern, and these organisms are non-motile, non-smoring, and they have a capsule around themselves which is made up of hyaluronic acid. So apart from producing an enzyme hyaluronidase, they also have a capsule which is made up of hyaluronic acid that prevents them from phagocytosis and helps in spread of the infections. And some strains of streptococci such as group B and D produces a polysaccharide capsules, whereas group C strains produces a hyaluronic acid capsule, B and D produces a polysaccharide capsules. And the best capsule demonstration can be seen only in the N cultures by a process which is called as Quellen reaction. Now coming to the cultural characteristics, how can we identify this organism in a cultural media, and what is the type of culture in which we can grow this microorganism? As we know, these organisms are aerobic and facultative anaerobe, so we have to make use of this culture that can allow the growth of the organism satisfactorily on the media. And the temperature that they require to grow in an artificial media is 37 degrees centigrade. That is, the optimum is 37 degrees centigrade, can vary from 22 to 40 degrees centigrade. centigrade. And these organisms, because they are pyogenic in nature, they are exact in their nutritional requirement. That is, you cannot grow them in the basic media. You have to provide special ingredients for these organisms to isolate these organisms in the artificial media. So this type of media that have special ingredients added to them are called as the enriched media. So the media, the basal media, can be enriched by adding blood, serum and other important factors that allows only the growth of these type of microorganisms. And the growth can be satisfactorily seen in the form of a media which contain carbohydrates or the blood or a serum enriched media. And in the blood agar, these organisms can grow very well if the blood agar has 5 to 10% of carbon dioxide. And after 24 hours of incubation, you can see the colonies in the form of small, which are 0.5 to 1 micrometer, and they are cellular 1 mm. Sorry, it is in the form of shape. They are expressed as micrometer, whereas in the colonies they are 1 mm, and they are circular. They are semi-transparent, convex disc. They show clear hemolysis because they are hemolytic organism. They completely rise the RBCs, and a zone of clearance is seen around the organism. It is called as a clear hemolysis, and it is the best method to isolate or identify the organism on a blood agar. And these organisms, they are highly virulent in nature and extracted freshly from the lesion. They usually form a type of colony which is called as a granular colony or the mat type, whereas the organisms that are of non-suppurative lesions usually forms a mucoid colonies. And here if they express a capsule, the mucoid colonies can be seen, and the presence of the capsule expresses their virulence nature, and the virulence nature organisms are similar to that of the mat type that we have extracted or isolated them in the media which contains the inoculated organism from the fresh lesion. And in the liquid media, these organisms show granular turbidity and powdery deposit, and no pellicle is formed. In the biochemical reaction, you can identify or differentiate streptococcus pyogenes from the other organism by a PBR test, which is a pyrrolidonyl naphthalamide test. This test helps us to distinguish between specific streptococcus pyogenes from the other hemolytic organisms. And the failure to ferment ribose is also another important characteristic by which you can identify these organisms with the help of biochemical test. Coming to the resistance, now this concept of resistance helps us to treat the organism. Once we know whether the organism is whether it is resistant or susceptible, you can go for that particular treatment. So resistance of the organism is very helpful for us to identify whether the organism is a multiple drug resistant organism or it is a sensitive organism towards certain kind of drugs. So these organisms, that is the streptococcus pyogenes, are highly delicate in nature. They can be easily killed on application of heat at 52 degrees centigrade, and they usually die when they are stored for a longer period of time, until unless they are stored in a media which is called as a Robertson's cooked media at 4 degrees centigrade. Otherwise, immediately when exposed to the external environment, these organisms cannot survive, and they can survive in the dust for about weeks together, and they are highly susceptible to different types of antiseptics. That is, antiseptics are the substances that usually inhibits the microorganism or kills the microorganism. So these organisms are sensitive towards wide variety of antibiotics or antiseptics. Because these organisms are involved in wound infections, antiseptics are usually inhibitory to these microorganisms. And they have an important characteristic is that they can resist crystal violet, that is an aniline dye that is used to stain the organism. So this particular characteristic that the organism is resistant to crystal violet can be used to make the media a selective one to grow only the streptococci, because crystal violet usually inhibits most of the group of microorganisms. So when we incorporate this crystal violet in a media, we can isolate only the streptococci group of organisms, because these organisms are resistant towards the crystal violet, and they are susceptible to wide variety of antibiotics such as the sulfonamides and other antibiotics, and differentiation is usually based upon the sensitivity to bacitracin. This organism is highly sensitive to bacitracin, and hence sensitivity to bacitracin also helps us to identify this organism, and this method is usually called as Maxstad's reaction that helps to isolate or identify the organism in the laboratory test. Coming to the antigenic structure of the organism, that usually elicits the production of antibodies which are protective in nature. So these organisms have different antigenic component that can cause different pathogenic infections in the human body. So before going on with the antigenic component, we should have a great knowledge about how the cell wall is usually made up of. So these organisms mostly are encapsulated. They have a capsule around the cell wall within which the cell wall is usually divided into three layers. That is the outer layer, the middle layer and the inner layer. First, dealing with the capsules, as we know that the C strains of streptococci contains capsule which is made up of hyaluronic acid, and B and D usually have a polysaccharide capsule. Now the capsular material that is enclosing the other cell wall component helps to prevent phagocytosis or escape phagocytosis. This is the reason the organism is able to survive the immune system and cause the infection in the body. So the capsulated organisms are highly virulent in nature and they can cause the infection in the body, and against these capsule antibodies are usually produced that are protective in nature. Next, coming to the cell wall. The cell wall of streptococcus is a bit complex when compared to the other gram-positive organism. Here the organism consists of an outer layer consisting of a protein and lipoic acid, the middle layer consisting of a group specific carbohydrate. So it is this region which contains a specific carbohydrate called as a C carbohydrate, and based upon this C carbohydrate presence or absence, the Landsfields had classified the organism into 8 different types. Now coming to the inner layer, which is made up of peptidoglycan or containing a mucoprotein that gives the cell integrity to the organism, and it is also the one that enhances the inflammatory response in the host once the pyogenic infection starts. And the main purpose of the peptidoglycan is not only to give cell rigidity and gives a shape to the cell, but also carries out certain biological properties such as pyrogenic and thrombolytic. Pyrogenic is fever inducing, thrombolytic is dissolving the clots of the fibrin that are usually formed during the particular type of infection. So this helps the organism to spread its infection from one part of the body to the other. Now, as discussed, serological grouping is mostly based upon the C carbohydrate that is present in the middle layer, and it was Landsfield who classified them into different types. And because the C carbohydrate is present inside, it is present internal, you have to extract the C carbohydrate to go for the classification of this streptococcus species. So the tests that are usually done to extract the C carbohydrate and go with the Landsfield classification is you can grow the organism in a specific media which is called as a TET headwit broth containing hydrochloric acid. This hydrochloric acid extracts the C carbohydrate, and this presence or absence of the C carbohydrate helps to classify the organism. Apart from that, you can go with the Fuller's method where formamide can be used, or can go with the enzyme extraction, and the enzyme that is used over here is extracted from the Streptomyces albus, which is called as a Maxstad's method. And next method that you can go ahead to extract the C polysaccharide is the autoclaving method, which was carried out by Rance and Randall method. Now the grouping can also be done not only in the liquid media but also can go ahead with the agar gel precipitation, and this organism will have apart from the C carbohydrate, they have different proteins that are associated with the cell wall of this streptococci bacteria. So the proteins, antigens that are present in the outer part of the cell wall, that is the outer layer of the cell wall, are of 3 types: M, T and R. So M, among which the M protein is the most important one and highly virulent in nature, and this is the one that can inhibit the phagocytosis in the organism, escape the phagocytosis and cause the infection, and hence it is said to be virulent in nature. And it is antigenic because it is able to produce the neutralizing antibodies, and with the help of this antibody we can diagnose this infection in the laboratory test, and the antibody antibodies that are produced are said to be protective in nature. And this heat MTR protein is a heat and acid stable, and the Lancet acid extraction is mostly used to identify or extract this M protein from the streptococcus species. And 80 different M proteins have been recognized in different groups of streptococci. Coming to the T protein, it is acid labile, and it is trypsin resistant, whereas here in case of M protein it is trypsin sensitive. The T protein is trypsin resistant, and hence the whole streptococcal T antigen is mostly used in the reaction such as precipitation or agglutination stage. So demonstration is mostly used in case of T protein is by the slide agglutination test, where the trypsin resistant whole streptococci are used in the agglutination reactions, whereas the M protein can be usually identified by the precipitation test. And here the streptococci are treated with the trypsin because they are resistant, and these streptococci are mostly used in latest agglutination test, so also called as passive agglutination test. Now next is the R protein. Very few species of the streptococci exhibits or have these R protein, among which the type 2, 3, 28 and 48 are the one that contains the R protein, and the groups among the group, the B, C and G group contains the R protein. Apart from these protein, the organism also have a non-specific M associated protein which is called as the MAP protein. Next, coming to the pili or the fimbriae that are present external to the capsule, which are covered with the lipotecoic acid. So the M protein and the lipotecoic acid components are associated with the pili, and it is with the help of this M protein and the lipotecoic acid and the pili, the organism is able to attach to the host cell, specifically the epithelial cell of the throat. So the presence of the M protein with the lipotecoic acid and the pilus helps the organism to adhere to the epithelial cell and initiate the infection in the respiratory tract. So the ligand over here is the epithelial cells of the throat. Now coming to the next important part of the streptococci group of organisms is a cross reaction. So as we have already studied about this cross reaction, it is a phenomena where when an antigen enters, it induces the production of antibodies. But these antibodies, instead of going and clearing that particular antigen, they go and interact with other structurally related antigens of the host, causing a damage to the host. So that is called as a cross reaction. So most of the component of the streptococci organism has certain antigenic substances that are very much similar to the human host antigen. So there is a cross reaction that causes the damage to the host. So the variety of antigens that are related to the streptococci group of organisms are, the first one is the capsular hyaluronic acid. So it has antigenic similarity with the synovial fluid of the human host. So antibodies generated against the capsular hyaluronic acid, instead of eliminating this organism, they go and attack the synovial fluid, causing the damage to this particular area. Apart from that, the cell wall protein, as discussed, there are three types of cell wall proteins, M, T and R, the M being the most virulent in nature. The cell wall M protein is having a structural or antigenic similarity with the myocardium. So the antibodies generated against this cell wall protein can damage the heart. Next is the group A carbohydrates specific to certain type of streptococci. They have a structural similarity with the cardiac valves, and cytoplasmic membrane antigens that are present in the organism share structural similarity with the vascular intima, and the peptidoglycan layer that is present in the cell wall, the interior to the cell wall of the organism, have structural similarity with the skin antigens, causing skin rashes in most of the pyogenic infections. So these cross reaction can also be extended not only to the acute streptococcal infection, but also can extend to the sequel of infection that is caused by the streptococcal group of organisms, that is the rheumatic fever and the glomerulonephritis, because they share structural components similar to the organism, and the damages over here is to variety of organs in the rheumatic fever, and in glomerulonephritis it is the nephrons that are usually damaged. Now, the pathogenesis of the organism is via the toxins and other virulent factors that contributes to the pathogenesis of the organism. So here the virulence can be brought about by the enzymes that are produced and mostly the exotoxins. So enzymes and exotoxins are the virulent factors that contributes to the pathogenesis of the organism. And M protein, as we all know, it is a specific protein present in the cell wall which is highly virulent in nature, can escape the phagocytosis and also can bring about the cross reaction. Next is the C polysaccharide component on which we have classified the organism into different landfill groups. It also has a toxic effect on the connective tissues. Now coming to the first toxin that is produced by the organism, most prominent one and most lethal one is the hemolysin. Hemolysin, the name itself suggests that it is the one that brings about the lysis of the RBCs. So there are two types of hemolysins that are produced by the organism. One is a streptolysin O and the other one is a streptolysin S. Streptolysin O is heat labile, whereas streptolysin S is heat stable. So they have got their own characteristic to bring about the pathogenesis. So the streptolysin O is usually in the inactivated form in the oxidized stage. It gets activated only when it is in the reduced form. So in order to isolate these toxin in the media, you have to add a reducing compound called as sodium bisulfite, so that the active part of the streptolysin O can be isolated. And this streptolysin O is heat labile and it is highly virulent in nature, which can bring about the cardiotoxic activity and the leukotoxic activity. And the biological action of this is it can cause a lot of damage to the RBCs present in the human host, bringing about the different types of reactions such as the respiratory diseases. They can cause the skin and soft tissues diseases, etc. And this being antigenic in nature, antibodies are produced against it, and these antibodies are called as anti-streptolysin O antibodies. And the presence of this anti-streptolysin O antibody helps in identification of the infection. So it is called as the ASO titer. So this ASO titer or the anti-streptolysin antibodies are usually inhibited by the cholesterol, but they are not affected by the normal serum. And the chemical nature of this antibody can also be modified by different components that are present in the bacteria. And hence classical method is not mostly used. Nowadays we have got serological tests to identify this anti-streptolysin O antibody. So with the help of serological test, we can know the titer of ASO antibodies. So the titer greater than 200 indicates a recurrent infection or the pyogenic infection with the streptococcus species. So in order to identify the streptolysin O, we can go for anti-streptolysin O antibodies, and the ASO titer helps in identifying the recurrent infection of the streptococcal species. Next is the streptolysin S, which is heat stable or which is oxygen stable, and it can also bring about the hemolysis. It is also called as streptolysin S because it is soluble in serum. It is protein but not antigenic in nature. That is, it does not induce the production of antibodies. And the convalescent sera is nothing but the sera obtained from the patient who has already recovered from the disease. So these patients usually have high titers of the antibodies. The sera, when it reacts with the antigen, it does not bring about any reaction. So there is no specific test to identify the streptolysin as hemolysin. So this can be easily inhibited by the serum lipoproteins. Next, coming to the other type of exotoxin, which is called as pyrogenic exotoxin, also called as erythrogenic, dick or scarlatinal exotoxin. So this is the one that usually brings about erythrogenic or erythromatous reaction, also called as a dick test. And erythrogenic means it is able to induce a fever and it causes skin rashes. So mostly this test is used to identify the scarlet fever in the children. So the blanching of the rash is nothing but when you press the rash with the help of a glass, that usually disappears. Apart from that, you can go for intradermal injection of the convalescent sera to identify the scarlet fever in the children. So this exotoxin is the one that usually produces the scarlet fever and can be used to identify the scarlet fever in the children who are suffering in the tropics. And the primary effect of this toxin is it induces the fever, and hence it is called as pyrogenic in nature. And And the organism that induces this type of fever is called as the streptococcal pyrogenic exotoxin or SPE. And there are three types of these groups of organism, that is A, B and C. A and C organism are the one that produces this exotoxin with the help of bacteriophage. So it is a bacteriophage that encodes this toxin, and the organism that have been lysed with the bacteriophage is only able to produce the toxin. Whereas the type B organisms have this type of production of the toxin in the chromosome, that is the genes that are present in the chromosome. So type A and C are bacteriophage encoded, and the organism has to be lysed with the bacteriophage, whereas type B organism themselves have a gene in the chromosome that produces this type of exotoxin. And sometimes these exotoxin can also act as super antigen in nature, similar to that of staphylococcus toxic shock syndrome toxin. That was a super antigen, or that induces the production of a non-specific immune response that can produce a variety of cytokines that can cause the inflammatory diseases, or can cause different types of damages to the host cell. So super antigens are the one that produces a drastic immune response that can damage the host cell to an extent that it can even lead to the death of the person. So this exotoxin can also act as a T cell mitogen that induces the non-specific proliferation of the T cells, and as we know, the T cells are mostly involved in the production of the cytokines. The cytokines in turn bring about the inflammatory response, and the inflammatory response is to an extent that it can completely damage the host. Apart from that, it induces the fever, and ultimately the tissue damage over here will be immunological reaction, because the cellular immunity is involved over here. It is the different cells of the immune system that can get activated and cause the tissue damage to the host. Next is the streptokinase, also called as a fibrinolysin. Streptokinase, it is the kinase that can kill the clots. The fibrinolysin is nothing but that is able to lyse the fibrin clot that is usually formed as a protective barrier during an inflammatory response. So whenever there is an inflammatory response, there is a fibrin clot that is formed that prevents the spread of the infection. Now these organisms are the one that are able to produce enzyme or exotoxin which is called as streptokinase that can break the fibrin clot. So they have the ability to lyse the fibrin clot and spread the infection. And how are they going to do this is they can activate a plasma precursor which is called as plasminogen, that is important in the formation of the fibrin clot. So they completely inactivate this protein, and they cause the lysis of the human clot and helps in the spread of the infection. So this streptokinase is also antigenic in nature, and it is the one that induces the production of antibodies. So antibody against the streptokinase can be mostly used to identify this exotoxin in the media. Next is the antibodies, streptococcal infection, and the biological role of this streptokinase is to break the fibrin clot and remove the fibrin barriers that are usually formed by the lesions that are present during the inflammatory response. Now, when there is a fibrin clot that is formed, it can be easily degraded by the streptokinase, and this clot, when removed, can help in the spread of the infection. And mostly this particular streptokinase is used in the therapeutic purpose, where they can be used in a myocardial infarction, that is in the heart attack, where a clot will be formed in the heart, the patients, and will not allow the diffusion of the blood to take place. So here, in order to remove that clot, we can make use of this streptokinase, and also used in the different thromboembolic disorders. The thromboembolic disorders are nothing but the clots when formed can enter into the bloodstream and can go and block any type of organs in the body. So these are called as thromboembolic disorders. So the clot can be removed with the help of streptokinase enzyme. Next important exotoxin is the deoxyribonucleases, also called as streptodornases. So they are the one, as the name itself indicates, deoxyribonucleases are the one that targets the DNA molecule, and here the depolarization of the DNA takes place, and the pyogenic exudates usually contain a large amount of DNA from the nuclei of the necrotic cells, and they are the one that can liquefy the thick pus that is present in the patient, and it is an important serious character of the streptococcus species that helps again in the spread of the infection. That is, when there is a thick pus that is formed, it will not allow the movement of the organism. So this deoxyribonucleases can break or liquefy the pus that is formed during an inflammatory response and helps in the spread of the infection. Again, this can be used in a therapeutic purpose and to liquefy the exudates in the patients of emphysema. And streptococcus and streptokinase are usually commercially available in the form of enzymes or in the form of exotoxins to go ahead with the therapeutic treatment purpose. And here in the case of deoxyribonucleases, they are also antigenic in nature, but the ASO titer does not give a correct value. So we have to go ahead with the demonstration of anti-DNase antibodies. Next, mostly deoxyribonucleases are 4 types: A, B, C and D, and the B specific one are the one that are usually involved in the human infection. So in order to diagnose this type of exotoxin, we can go with anti-DNase B antibodies. Next is nicotinamide adenine dinucleotidase, that is NAD, which acts upon the coenzyme NAD and liberates the free nicotinamide. So nicotinamide adenine diphosphate is the one that is involved in the electron transport system, carrying and transporting the electron from one system to the other system. The physiological property of how it brings about the pathogenesis is not known, but it is said that it is highly leukocytic in nature, and it is also again antigenic in nature, which can be neutralized by the antibodies. Hyaluronidase is another important type of exotoxin that is produced by the organism or the virulent factor associated with the streptococcus group of organism. Hyaluronic acid is nothing but the intercellular cementing material that is present between the cell. Now the hyaluronidase is the enzyme that has the ability to break down the cementing material and spread the infection. So the organism has 2 types of hyaluronic or hyaluronidase that is associated with the organism. One is a capsular hyaluronic acid that is present enclosing the organism, and the second one is it is able to elaborate or produce hyaluronidase enzyme. So both of these are helpful in spreading of the infection from one part of the body to the other. And here the hyaluronidase stains that produces large amount of capsule that is hyaluronidase enzyme are usually the non-capsulated one, and they belong to the M type. And the enzyme is again antigenic in nature, where you can see the presence of antibody against this hyaluronidase or the capsule that is made up of hyaluronic acid. Next important concept is the serum opacity factor that is present in the organism, and the organism of the M type have the ability to produce certain lipoproteinases that can form opacity in the medium containing hordes or the swine serum. So the presence of this opacity helps in identification of the organism in the lab diagnosis. Apart from these exotoxins, they are able to produce different types of enzymes such as proteinase, phosphatases that can rupture the phosphate groups that are present in the human host, especially the cells containing the nucleic acid, and the esterases, amylases, and acetyl glucominidases and other toxin and enzymes. Now coming to the pathogenicity, how the organism is able to induce the infection in the human body. So because this organism is able to spread from one part to the other and has the ability to target different organs in the body. So first we'll begin with the respiratory tract, where the organism is able to establish itself and cause the infection. So mostly these organisms are pyogenic in nature, causing wound infections, and they can spread to different parts via the lymphatic system or the blood circulation system. Now first coming to the respiratory infections, here the primary infection or the primary target that is seen over here is the throat. Because these organisms are the normal commensals that are present in the throat, they can bring about they can carry out the opportunistic pathogenic infections related to this organism. And the first symptom that can be seen with this organism is a sore throat, and it can cause tonsillitis and the pharyngitis. Usually tonsillitis is seen in the young people, whereas the pharyngitis is seen in the children's, very small children's. Next is this respiratory infections. Now, how is the organism able to attach to the throat and cause the infection? So, the organism consisting of lipoic acid along with the M protein and the pilus binds to the pharyngeal epithelium, and the ligand here is the glycoprotein fibrinolectin that is present in the epithelial cells, which where the organism with the lipoic acid usually goes and attaches and helps to initiate the infection. It is said that this can also be initiated by the hypersensitivity reaction, that is a local denaturation or the local inflammatory response that is induced over here to cause the pathogenesis, can also be due to the hypersensitivity. And once established, this organism has the ability to spread to different surrounding areas and cause the infections of that particular organ, leading to otitis media, mastoiditis, and quinsy, Ludwig's angina, usually involving the skin of the mouth, that is the floor of the mouth, and the suppurative adenitis. And mostly these type of infections lead to pneumonia and rarely to the meningitis. And the pathogenesis can be enhanced by the different viral infections too. That is, once there is a viral infection caused in a person, that can bring down the immune system, which makes this streptococcal infection a secondary infection and can enhance the pathogenesis of this particular infection. So mostly when the person is infected with influenza or any viral diseases, he is more prone to infection by the bacterial diseases, mostly by the streptococcus species. Next type of diseases associated with the streptococcus is the skin and the soft infections. Mostly the target over here is the skin, and the soft tissues or the tissues that are related to the skin. So the organism is involved in the infection of the wounds or the burns, etc., and it can lead to predilection to lymphatics or the cellulitis, and the infection which are seen in the minor abrasion can lead to fatal septicemia. And two types of skin infections are usually associated with the streptococcus species. One is called as erysipelas, and next is the impetigo. Erysipelas are superficial lymphatics, and you can identify this with the help of certain characteristics on the skin. That is, the skin will be red, swollen, and it has an indurated demarcation. It is very rarely seen in the people, but old people are more prone to this type of erysipelas. Next is the impetigo, which can be seen in the young children. Usually the ones that are 5 to 8 years or below 5 years. And the type of protein that is involved here is, or the species involved here is, which contains higher number of M types, whereas lower number of M types are involved in the respiratory infection or the throat infection. Higher number M types are involved in the impetigo. And impetigo and scabies lesion together can lead to acute glomerulonephritis in the children in tropics. And the antibodies produced against this type of diseases are anti-DNase and hyaluronidase antibodies that helps in identifying the infection due to impetigo. And next is this can lead to toxic shock syndrome. So most of these organisms are also called as the flesh-eating bacteria, because they elaborate a high amount of hyaluronic acid and helps in the spread of the infection. And they are the one that can cause the lesions to an extent that it completely damages the area wherever they are usually growing and producing the exotoxin. So toxic shock syndrome, similar to that of staphylococcal toxic shock syndrome, toxins can also be produced by these organisms, mostly of the M types. Next is the genital infections, which are associated with the streptococci. Mostly this organism is a normal flora that can be seen in the female. So the aerobic and the anaerobic organisms are the normal inhabitant in the female genitalia, and it was thought to cause a puerperal fever, which is also called as a childbed fever. Initially the puerperal fever was thought to be exogenous in nature, and which led to more mortality in the death of the child or the newborn child. So in the mortality rate was brought down by Semmelweis or Ignaz Semmelweis, who who said that by washing hands we can reduce this childbed fever. But nowadays we come to know that this popular fever is not due to exogenous infection, but it is because of the endogenous infection, as the female can harbor this organism in the genitalia as a normal flora and can sometimes cause the infection. So endogenous infection are mostly because of the anaerobic group of organisms that inhabit the female genitalia. And the treatment of this particular disease was not possible before the discovery of the antibiotics. It was only after the discovery of the antibiotics that this particular type of infection can be overcome. Next is the upper suppurative lesions associated with the organism. They can cause abscesses of the internal organs such as heart, kidneys, brains, etc. And they can also cause septicemia and pyemia, that is pus forming. Coming to the non-suppurative infections or the complications, which are nothing but a sequel to the acute streptococcal infection. That is, once a person has been attacked with this streptococcal infection, there is a sequel to it that usually follows in very few of the patient, which leads to rheumatic fever or the glomerulonephritis. So once the acute infection subsides, it is after first to 3 weeks that you can see the sequel that is following the acute streptococcal infection as a rheumatic fever or glomerulonephritis. So here in the rheumatic fever, the lesions that are usually associated with the rheumatic fevers are carditis. It can cause connective tissue degeneration of the heart valves or inflammatory myocardial lesions which shows Ascoff's noodle. So demonstration of the Ascoff's nodules can help to identify the rheumatic fever, which in turn helps to identify the streptococcal infection. And here we have a strong antibody response where the antibodies are generated against this the component that is a rheumatic fever can be easily detected during this particular disease. And one of the reasons that is associated, or the localized reason that is associated with the non-suppurative complication, is because of the hypersensitivity reaction and certain component of the autoimmunity, and most probably it is a cross reaction. And here the cross reaction usually takes place between the M protein of the streptococcal species with the myocardium of the heart, and the antibodies that are generated can damage the myocardium and cause the rheumatic fever. So cross reaction is the most important reason that can be attributed to the non-suppurative complication of rheumatic fever. Next is the nephritis. Nephritis is not as a rheumatic fever can be caused by any strain of streptococci pyogenes. Here nephritis is not caused by all strains but only few strains which are called as nephritogenic in nature or nephritogenic types, and the skin infection are more important over here and can be easily identified with the help of skin infection. And this nephritogenic or nephritis is self-limiting episode. It only happens for a particular period of time and there is no reinfection, whereas for rheumatic fever there is a reinfection that can completely damage the heart of the patient. But here in case of glomerulonephritis, it is a self-limited episode without any reinfection, and the pathogenicity is usually due to the cross reaction between the glomerular membrane of the kidneys and the M protein again associated with the streptococcus species. So the antibodies generated towards the organism can damage the nephrons and can ultimately damage the kidney. So apart from that, the other reason that can be given for this is immune complex disease. As we have seen in type 3 hypersensitivity reaction, the formation of immune complex can also lead to glomerulonephritis, where the complex goes and hides themselves inside the glomerular membrane, and during the immune response, during the extraction of the immune complexes, there is a large inflammatory response mediators that are released that can ultimately damage the glomerular membranes. Coming to the epidemiology, how this infection is spread and what are the sources that helps in the spread of the infection. So it is the source of the infection are the humans which carry the organism in the upper respiratory tract. This can be a patient or a carrier who can harbor the organism, and sometimes the patient can be symptomless, and these symptomless carriers can also transfer the infection, and the transmission is usually done via direct contact or using the fomites, fingers or vessels, etc. And mostly seen in the tropics. These organisms can cause the infection during the winter season, and in the tropics they can be spread by non-biting insects such as the ice gnat of Hippolaites, and mostly seen in the winter season, attacking mostly the 5 to 8 years of children, and tonsillitis being seen in the adults, and the pharyngitis mostly seen in the young children. Crowding is another important factor for the transmission of the disease, and the immunity associated with this is a type specific immunity, that is the antibodies that are generated against the different components gives a protection against this particular organism and also helps in identifying the infection. So it is a type specific immunity, whether it is a humoral or it is a cell mediated, mostly it is a type specific, and antibodies are mostly involved in protecting against this type of diseases. Coming to the lab diagnosis, how can we identify this organism? So once the patient is suffering, the best part to collect from the patient to identify the organism is the sputum, and in case of acute infection, it is a culture method is advised to identify the organism in the culture, whereas in the non-suppurative lesions such as rheumatic fever or glomerulonephritis, you can go only with the antibodies, because you cannot identify the organism. Once you know the organism, you will not know whether it is involved in rheumatic fever or glomerulonephritis. It can be different reason by which this non-suppurative lesions can be induced. So for non-suppurative lesions, you can go for antibodies, and for acute infection, that is the wound infection or the pyogenic infection, culture is the best method. First thing that we do is go for the that is a microscopic testing, go for the gram staining, where the organism can be stained in the pus or the CSF, that is the cerebrospinal fluid. If there is a localized infection, pus is the preferred choice, and if it is a systemic infection, you can go with the cerebrospinal fluid. So gram staining gives us an indicative of the organism, that is the presence of purple or the violet chain cocci in the slide helps us to identify the organism as streptococci. And next thing is, you can grow in the culture. The culture that we are going to use, or the media that we are going to use, is an enriched media. Mostly the blood agar is used, and the organism has to be collected aseptically under a supervision, because most of the organisms are the commensals one. So we have to identify the pathogenic group of organism from the commensals. So immediately that has to be transferred onto the media, or if it is taking time, then go for the transport media, which is called as a Pike's medium. The Pike's medium is nothing but a blood agar containing 1 in 10 lakhs crystal violet and 1 in 16,000 sodium azide. It helps the organism to maintain its condition till it reaches the laboratory. Once it reaches to the laboratory, they are inoculated onto the blood agar, plated at 37 degrees centigrade or incubated at 37 degrees centigrade anaerobically, and the blood agar should contain 5 to 10% carbon dioxide. And the blood that is used to prepare the blood agar is mostly from the sheep blood agar, because in the other type of blood samples, there is a possibility that can be contaminated by Haemophilus hemolyticus organism. So this can cause a confusion in the diagnosis. So go for only the sheep blood agar to identify the streptococcus group of organism. And once the organism has been identified, then you have to go with the Lancefield technique for grouping the organism, and apart from that, you can go with the fluorescent antibody technique. And max test, bacitracin test is mostly employed to identify the organism in the media. Here in the bacitracin test, what we do is, as we know that the organism is sensitive to bacitracin, a filter paper disc that is impregnated with bacitracin is placed on an inoculated plate containing the organism on a blood agar and incubated. A clearing zone is seen due to the sensitivity of the organism. So this helps in identifying the organism, because bacitracin is highly sensitive to organism is highly sensitive to bacitracin. Next is, you can type the organism for epidemiological purpose, and this can be done either by the precipitation or the agglutination test. And nowadays we have a rapid diagnostic test where we have a kit containing streptococcal group A antigen that has been prepared from the throat. So we do not go for all the culturing technique to identify the organism. Go for the kit test to identify the organism, which we can get the result within 1 to 4 hours. Next is for the rheumatic fever and the glomerulonephritis. As already discussed, you have to go for the sera, anti-sera, that is the antibodies that are produced against these type of infections, and mostly the anti-streptolysin O antibody titration or ASO titer is mostly used. So greater than 200, ASO titer greater than 200 indicates the persistent or recurrent infection with the streptococci. But in case of glomerulonephritis, the ASO titer is very less. So in that case, we can go with the anti-DNase B antibody or anti-hyaluronidase test, mostly to identify the pyoderma, that is involving the pyodermic infections. Apart from that, you can also go with the streptozyme test, which is also called as a passive slide hemagglutination test. So here we are not making use of a direct agglutination test. Passive is nothing but sensitizing the RBCs with the crude extract of the enzyme that belongs to the organism, and making use of this RBCs for the agglutination reaction. So that is a streptozyme test. Streptozyme, thus the enzyme from the streptococcus organisms have been used to sensitize the RBCs, and these are further used in the test. That is called as a passive or a latex or a passive slide agglutination test. Coming to the prophylaxis, prophylaxis over here is mostly in the prevention of the rheumatic fever in the children, and the penicillin is often administered for a long term in the children who have been diagnosed in the first stage of rheumatic fever. In order to prevent them further damage to the heart, you can go with the penicillin administration. And here the streptococcal reinfection can be inhibited, and damage to the heart can be inhibited by the penicillin. Apart from that, you can also go with erythromycin. That is the one that if the person is allergic towards penicillin, can go with the erythromycin. And in case of glomerulonephritis, the other antibiotics can be used, but sulfonamides cannot be used for the treatment of this disease. And treatment is mostly by the penicillin G, erythromycin or cephalexin. Allergic individual can be given with erythromycin or cephalexin, and there are no antibiotic drugs that can have effect on the rheumatic fever and the glomerulonephritis. Only prevention can be done. Next is the other group of streptococci or the hemolytic cocci. As we know that there are different groups of streptococci organism, and we have first classified them based upon the hemolytic pattern, and then with the Lansfield group of classification. So apart from Streptococcus pyogenes, there are other hemolytic cocci that are involved in human infection. Mostly they cause infection in the animals, but sometimes they have been associated with the human infections too. So these streptococci of the group B, C, D, F, G, and rarely you can see H, K, and O that causes the human infection. So starting with the group B, it is the one that was mostly involved in the cattle infection causing bovine mastitis, but it has gained a clinical importance when it was first isolated or first seen in relation with the neonatal meningitis. So it is the one that was causing the meningeal inflammatory diseases in the neonates who was born from the mother during the normal delivery. So here the infection in newborn can be divided into two stages. One is the early stage, next is the late stage. Early stage is within a week you can see this type of inflammation or inflammatory reaction leading to the septicemia, meningitis or pneumonia, and it is during the maternal birth, the vaginal birth, you can see that the organism getting established on the newborn child and causing the infection. Now coming to the late onset type, it is mostly environmental in nature but not due to the mother, and the group infections that are related here in the neonates are arthritis, osteomyelitis, omphalitis, conjunctivitis, respiratory infection and peritonitis. And here they can be identified easily with the help of a test which is called as hydrolysis of epurate, which is a presumptive identification of this group B type of organism. Can go with CAMP, Christie, Attack, Munch, Peterson reaction to identify this organism in the laboratory. And most of these organisms have a capsule around themselves, and these capsules are mostly made up of polysaccharide which are virulent in nature. And this organism contains 9 capsular serotypes of organisms. Coming to the group C, here we have the organism mostly involved in the animal pathogens, and they are usually 4 types: A, B, C and D. And the one that is causing human infection is streptococcus equisimalis species, mostly causing the upper respiratory tract infections, and also involved in the abscesses of endocarditis or osteomyelitis or brain abscesses, pneumonia and puerperal sepsis. Here the organism can be treated with the other forms of penicillin. Because this organism is penicillin tolerant, we can go with gentamicin, and the organism can be identified by a biochemical reaction, which is the one that it has the ability to ferment hallose. And streptolysin O and streptokinase are usually extracted from this type of organism that are mostly used for the therapeutic purpose. Next is the group F organism. They are the one that cannot be easily grown on a blood agar until unless they are provided with a good amount of carbon dioxide. Because they form small colonies on the media, they are called as minute streptococci, and mostly involved in the suppurative lesions, and they have a special protein associated with them which is called as the streptococci Mg protein. And towards this protein antibodies are usually produced, and this Mg protein or alpha-lytic strains are the one that are involved in atypical pneumonia, and demonstration is with the help of agglutinins. Group G, they are the commensals, and as already discussed, these can cause infection given an opportunity. When the immune resistance comes down, they cause the infections. Commensals of the human, monkeys and dogs, and they can cause tonsillitis or pharyngitis or endocarditis. Usually following the cardiac lesion, they can lead to endocarditis. Group H and K are the one involved in infective endocarditis. Group O causes acute tonsillitis, and group R mostly in the pigs. So people who are in very close association with the pigs are usually affected with this type of R strains, and they are the one that causes meningitis, septicemia and pneumonia in the patients. Now coming to the group D, Enterococcus species. So these type of organisms are the one that are non-suppurative, and they have been initially classified into Streptococcus group of species, but later on, because of certain special characteristics associated with them, they have been classified in a different genus called as Enterococcus. So they contain different types of species called as Enterococcus faecalis, Enterococcus faecium, Enterococcus durans, and the non-Enterococcus forms are Streptococcus bovis and Streptococcus equinis. And here the organism can be identified based upon the chemical background, and mostly the Enterococcus faecalis are the one that are the one causing the infection in the humans, involving the intestinal tract, and they can ferment mannitol, sucrose, sorbitol and esculin, which is a presumptive test for identification of the organism based on their biochemical reactions. And they have the ability to form black colonies on the tellurite blood agar. And the enterococci are usually the inhabitants of the skin, genital tract and the saliva, can cause various genitourinary infections, or you can say gastrourinary infections in the patient. Next is the non-enterococcal group, mostly in the one that are penicillin sensitive, and they can grow in a salt concentration of about 6.5%, and they are the one that can satisfactorily grow on a media containing bile, and they are the one involved in urinary infections. The last one is the viridans group, that is also called as a miscellaneous group of streptococci. So they are also called as the Streptococcus viridans, which causes green coloration on the media, and they are the one that are present in the respiratory tract, mostly the mouth, upper respiratory tract, normal flora of this particular area, and causes the alpha lysis viridans and viridan group of organisms they are called as. Examples of the organism or the species that are associated with this are Streptococcus mitis, Streptococcus mutans, Streptococcus salivarius and Streptococcus sanguis, and they are mostly non-pathogenic in nature, and sometimes they can cause bacterial endocarditis by Streptococcus cereus. Now, during the tooth extraction or dental procedure, they are the one that are the cause of bacteremia and can cause the secondary infections in the dental decay, and transient bacteremia can be seen after the tooth extraction and the dental procedure, and they are the one that can get implanted on the damage cause the damage to the prosthetic valve or the transplanted organs, and once they get implanted, they are able to divide and form the vegetative form and can cause the infection in that particular area wherever the implantation has taken place, and hence prophylactic antibiotic therapy is given before the tooth extraction or the dental procedure that is taking place. Now, coming to the streptococcus mutans that is involved in the dental caries, this organism is the one that is usually seen as a normal flora. Teeth will not allow the establishment of the organism because of enamel that is smooth in layer. So, this streptococcus mutant has the ability to convert the confectionery sugars into acid. Now, once the acid is formed, it can etch the teeth, and it helps in attachment of the organism to the teeth. Now, during this procedure, it produces an acid that can damage the dentine, and it produces the dextran. Along with this dextran, the food mucus and along with the other organism lead to dental plaques, which can further be carried on to dental caries. So, these are the group of streptococci organisms that are mostly pyogenic organisms and causing different types of infection in the body, attacking different organs of the body.